Hello. Hi, everyone. I think we are getting ready for this afternoon's event. Welcome here, everyone, and welcome to this Lunch and Learn event hosted by Biointron and Mass Bio. And I'm really pleased to be here. Today, we're going to have a couple of distinguished speakers from our collaborator and our longtime customer to give you some insights from their view of the industry. And my name is Lei Shi, and I'm currently the Senior Vice President of the company of Biointron and responsible for all antibody discovery related issues, services, projects, and business development, helping our business development colleagues in both the US and all over the world. And just a brief introduction myself, and I have more than 20 years of experience in early discovery. And first, I was actually here in Boston for my postdoc study at the Mass General for 5 years, studying innate immunity at Alan Ezekowitz's lab. And later on, I joined Johnson & Johnson in Pennsylvania, working on a novel P9-based phage display technology. And some of them, actually last night, I met a customer and he was working for MorphoSys, and I was laughing. So we were trying to replace MorphoSys phage display system at the J&J, and I was the one leading the project. And after almost 10 years at the J&J, I joined a series of startup companies in Shanghai, first working on GPCR targets, and we delivered one of the very first functional antibodies against cannabinoid receptor 1. And later I joined a couple other startups working on bispecifics and ADCs. And all through that period, I became one of the first customers of Biointron from 2016, when the company just started to offer a revolutionary service that shortens the whole expression and purification time from 7 to 8 weeks to 2 weeks. And ever since then, I have been a customer of Biointron, and later on decided to join the company more than 2 years ago, and now leading the discovery effort of the company. So with that, I'm going to actually introduce our speakers later on, but we have Dr. Chimi Wang here today from Saijin, and also Dr. Hitesh Soni from Tectonic Therapeutics, and I will give you more details after that. And now it's the hard advertising time, and I'm going to tell you a little bit more about our services. All right. I see many new faces, and some of them are our old friends, but the majority of you may not be aware of who we are. And so let me just tell you what about the Biointron. So we are a company that established in 2012, and our CEO and the founder is actually joining us straight from the airport this afternoon, and he just flew from Korea from a conference, and Mr. Zha, and he started as a company working on recombinant proteins, but then Biointron catched a trend and opportunity in around 2016, and started provide the service in antibody production. And we now have about more than 600 people with a facility of more than 28,000 square meters, and we actually just started to put a new production line in a brand new building consisting of four buildings, and we in the past few years we have served and maintained a customer base of more than 2,000. And so our headquarters is actually in China, Shanghai, China, and we have a production center about 2 hours away from China. And our international presence has been exemplified by the U.S. Business Shipping and Marketing Center here in Boston. And we also have a European logistics center and handling all the shipments for our customers. And our international BD head, Boris, many of you probably are familiar with, are the first person that you get to know from our company. And he's showing us today as well. And the brief history I just told you about this. And so from in the past decade, we have grown tremendously from a small workshop to a company that we deliver more than 100,000 antibodies every year. And we did just this year, we filed for IPO with the purpose of getting more resources for future expanding and the growth. So you can see that from 2021, our growth has been really rapid, and we have a forecast for this year about more than $70 million of revenue, and we have served many customers from 26 countries and the regions, and including the majority of the top 20 multinational pharmaceutical companies, such as Johnson & J&J and AstraZeneca. So, and locally, Moderna has been one of our major customer as well. So, this is the production facility you could find. You will see if you have a chance to visit us, and we have to support this large numbers of antibody production and discovery projects, and we have a clean room with a molecular lab for DNA sequencing with more than 20 DNA sequencing machines. We have, along with plenty of shakers, more than 300 of them. And the bioreactors can handle from 15 liter to 200 liters to support our service. Not only that, one of the hallmarks of our service is we achieve this levels of capability by doing a lot of employing a lot of automated workstations. That's mostly custom built, and so that we can make the whole process from gene synthesis to plasmid purification to sample prep and allocating to labeling, everything you can think of along the process of antibody expression production can be used can use automation in some way, and this is how we keep innovating ourselves and improving the whole process. So and most recently, we also have procured a large number of instruments that we are moving from a mass production and high throughput antibody production to high throughput analytical capability, and we have 3 vehicle AK as well as a Katara, the high throughput SLA, and we have recently also have the IQ screener, DLS play reader, DSF, nano DSF, and TET. All of these instruments are supporting our goal for developing new assay development as well as the developability study capability. And I'll tell you a little bit more later on. So with that, what do we exactly do? And you probably, I mentioned antibody production, and this is absolutely the main business of our company. and we've been making IgGs, antibody fragments via churches, and most importantly, the industry is moving from more and more from monoclonal antibodies to bispecifics. We also make a stable cell lines for our customers. And there's also a rapidly growing business in antibody discovery using two major platforms, which I will introduce to you on both phage display and single B cell screening. Those are fascinating and high-speed discovery platforms that we hope would further support our growth and also accelerate your discovery need. We also do a lot of antibody addition, humanization, and affinity maturation. These are something fully utilizing and taking advantage of our high throughput production capability. Cell line development I just mentioned, and we are now making more and more efforts in the in vitro assays that provide a high value services beyond just antibody production and purification. These are becoming more and more interested in, and demanded by our customers. We also have a catalog of product services that provide many of these common target positive control antibodies for your in vivo studies. These include the mouse and anti-mouse target antibodies that and biosimilars and control IgGs and everything that's you can imagine that can be readily used in your lab for your in vivo model study. And let me just give you a quick review on the expression business. This is by far the most important business that the Biointron offers. Like I mentioned, we delivered more than 100,000 antibodies last year, and the number is growing. Not only that, we could handle up to 3,000 per batch of the antibody and support those high throughput demand. Many of these customers are doing a lot of AI aided research, and they want to generate as many possible samples as they can and have a quick readout of how these molecules behave. And we can not only go from a small scale, but of micrograms of antibodies, but we can also go all the way up to gram level of production. And with the majority of the mAb services and can be done from gene synthesis, meaning that you send us the sequence of the antibody that you want to make, and we deliver purified products in 10 to 14 days. And that's been the hallmark of our service that differentiates us from other competitors. And I see a lot of companies try to catch up, but we are making progress. We are optimizing. We are innovating ourselves all the time, and we will keep our competitive edge along with the quality service product. So just give you an example. So this is one of the early example of more than 800 IgG was asked by our customer, and we actually delivered them in one batch, in a single batch in 10 days. And the yield distribution, you can see that it's around, most of them is above 100 meg per mil yield of titer, and with the vast majority of them reaching 95% purity. So this is just one of these examples. Not since monoclonal, we started with the monoclonal antibody production, but more and more bispecific has been demanded, and we actually see almost half of the orders are coming from customer want to make a bispecifics or multi-species. And interestingly, we have so far accumulated experiences in more than 260 formats coming from all kinds of customers. So you can imagine our scientists, our customers are really imaginative in terms of how they design these bispecifics. And these can be also made as fast as 2 to 3 weeks. Of course, by specific, most likely it will take a little more time or exploration in research, but we are we are also trying to make it fast. Just give you some example of some very popular formats of the IgG of the bispecific, including these symmetric IgG single chain V formats that can be purified in one step, reaching almost reaching more than 95% purity. And I mentioned the scale up production, and this has been also growing to, because many people start screening using small samples, and then later on we found that they are moving towards downstream and making samples for animal studies, for preclinical tox studies, and this is where we can also come in to produce large amounts of large quantities of antibodies. That's the main business that we talk about, and then I want to quickly go through some of a couple of the discovery platforms. The alpaca, we use alpaca VHH as our main antibody discovery platform. The reason being VHH antibody support are more and more popular, and in the world of new era of bispecifics, and the VHHs are a lot easier to handle. They are stable, they're modular. You can easily put them together to form bispecifics or multi-specifics, and they are also as stable. They could be as stable as IgG. So when you put a proper module like an FC as a fusion partner. So we have our own farm of more than 300 naive animals that we started feed those baby alpacas from 4 months and until they are mature enough for immunization. That way, we keep track of all the growth records, making sure they are healthy, they are naive, and they are free of any other immunogens. And a lot of our customers have a high standard of using naive animals. And we have delivered, since then, we have delivered last year more than 120 discovery projects for our customers. And if you ask me how these projects behave, I can give you an example of about 30 projects from one single customer. And this is a non-scientific statistics because these are we collected 600 more than 600 antibodies out of these discovery campaigns. Actually, this number is coming from more than 2,000 initial hits, and these are the ones that are most chosen to move forward in more detailed affinity study. So you can see that the majority of the clones are having animal level affinity, and have a healthy distribution of affinity binding curve, binding data, and ranging from sub-nanomolar close to picomolar and to some of the more weaker ones. And I mean, it depends on your purpose of the project. So it's not necessarily the higher the better for the affinity, particularly for something of the same, right? I've seen many people asking for a range of different affinities and as a specific interest. So And not only that, we now have a VHH antibody naive library that consists of more than 300 animals, the cells from the DNAs from more than 300 animals, with a total size of the library reaching to more than 10 to the 11. And this library has recently been validated with more than 10 different targets, including different types of like a soluble ECDs, membrane, single pass membrane targets, multi-pass, GPCRs, and we are pretty excited that this could be a very good complementary capability to our existing immunized library like that. So this is a particularly useful for targets like difficult to immunize with a high homology, maybe with alpacas and mammals and or protein complexes, which cannot be used for immunization very well. So I think that this will be a very good tool. The second single B cell, the second discovery platform I'm going to mention is our single B cell platform we call the AppJob, and this is particularly important because today Qiming is going to tell you more about how the collaboration between Biointron and the Sargent was started, and how we go. How are we moving forward towards a more powerful discovery platform in taking advantage of both AppJob as well as the scientists humanized and platforms. So stay tuned for that. But let me tell you, our single B cell technology is based on microfluidics using droplets technology, and it's been used very successfully for generating mouse or rabbit antibodies, different types of antigens. But the most important feature of this platform is we can deliver the verified leads and antibody leads from immunized mouse, from immunized animals to verify these within a month. I mean, 2 to 3 months. That's including the immunization time, and this is a month shorter compared to the traditional hybridoma screening method. And our droplet-based platform is such that you can view this as using 2 liquid phase. So one is the aqua phase that contains all the reagents, including antigens, binding buffers, and single B cell, single plasma B cells that we enriched previously, harvested from the animal, and then it's been wrapped up by a oil phase to form a droplet with a single B cell in it. And then after incubation of a little bit time producing to allow the cell to produce a minuscule amount of antibody. And then we can use FRET technology to detect the binding of the antibody with the antigen and sort those positive cells out, and using NGS sequencing and the barcode with the barcoded primers for both heavy chains and light chains, we can then obtain thousands of sequences, all paired, or naturally paired, and from that we will use our high-throughput antibody production platform to make small samples for ELISA or more detailed SPR or BLI affinity testing. So with that, you can actually process millions of plasma B cells in one day. and sort out those possible positive cells and get through the NGS sequencing in a week to get all naturally paired antibodies. And within a month, you will get a purified samples with a confirmed binding data. So this is fully taking advantage of the droplet technology, single B cell sorting, plus our high throughput antibody production to accelerate your discovery timeline. And it's also a very powerful tool that you can get as many as antibodies you can get from one single animal versus I've been using hybridomas, and the throughput for hybridoma is very limited and also very labor intensive. So quickly go through the humanization of the affinity maturation. We use our high throughput 2-week scheme of antibody production to with a humanization of the CDR grafting based humanization design, and we will get a humanization data in a month or so, and plus the affinity maturation platform is also taking advantage of the high throughput gene synthesis and production, in which that we actually we mutate each CDR positions with a 17 saturation mutagenesis, and then produce each one of them in the high throughput format, and test the individual clones and identify those clones with elevated or, in other words, decreased affinity to find those so-called hotspots in the CDR region, and then combine them to form a secondary round of mutants that could elevate the affinity in like dozens or more than 100 fold improvement of affinity. So these are some of the examples of affinity maturation and humanization. And I'm not going to go through this, and we have flyers, and we have our colleagues can tell you more about this. I'm going to just touch upon the in vitro assays. There's a lot of functional in vitro assays we do: internalization, survival, killing, T-cell based activation assay. These are very routinely done, along with the effective function assays that we've been doing for many of our customers. And not only that, so you can see that this slide gives you an overall view of what we can do to focus on the discovery phase from the reagent generation to cell line development, what we can do along the way to support your early discovery projects. And that's all empowered by our high throughput antibody production platform, because at every stage, every step along the way, you will need a lot of samples to be made and then made quickly to assess these properties. And this is the current state, but in the next 3 years, we are enhancing our service capabilities by putting new assays, new capabilities like the membrane protein expression, new libraries like naive libraries, human naive libraries, alpaca naive libraries, humanized alpaca libraries, humanized VHH libraries, as well as a lot of assays that to deepen to get more functional data from your projects, and also give an opportunity to have an early peek of the developability properties of your kits. And this, you can see that there's a big stars there. Yes, I think that this will complement and make the whole chain more complete for our service from antigen prep to cell development. And there's a lot of exciting things that's currently happening, and I hope that I will have a chance to tell you more in the next event. So that's all from me. That's very much for listening. So now I think it's my honor to. Okay, so now it's my honor to introduce our distinguished speakers, and the first one is Dr. Chiming Wang, and he's now the director of product strategy and this strategy partnerships at the Ciogen. He got his PhD locally at the Tufts, and he's really a truly immunologist, I can tell. Unlike me, I was a molecular biologist and then transitioned to innate immunity, but he's been studying adaptive and innate immunity from the very beginning. And he's also been focusing a lot of efforts on the autoimmune diseases. He later joined the Jackson Lab, and I think he he joined the Ciogen a couple years ago, right? 6 months ago. Wow, it feels like a long time you've been here. And he's gonna tell you more about the Ciogen platform and how the collaboration was formed. And please.
Thank you, Lei, for the very generous introduction, and thank you everybody for coming. And it's really an honor to come back here where I got my PhD from and talk to you about some of the work we do at Ciogen. So I'm an immunologist by training and / mouse geneticist by training. So a lot of the stuff I'm talking about is more on the mouse platform that we provide or we partner with Biointron to accelerate antibody discovery. I think this slide really sort of like captured the core strengths of these two companies. So we are focused on the providing the animal models or humanized models, and then it is in partnership by Intron's expertise in antibody discovery and antibody expression. So ultimately, we wanted to translate it into therapeutics that can ultimately benefit human health. So what is what we call HUGO is really an acronym for Humanized Genomic Ortholog for Antibody Development. It's really a umbrella program for 3 platforms we currently offering to the market. One is what we call HUGO Map, which is a conventional H2L2 antibody platform, and you can see that the variable region is humanized. And so in this mice, it provides an antibody of human variable region. And then we also have this, what we call HUGO Light, which is a platform for common light chain as one of the platform to provide bispecific antibodies. And last but not least, we also have the HUGO Nano antibody. So essentially by knocking out the CH1 domain of the heavy chain and knocking out both the kappa and lambda light chain, the antibody provides producing the mouse will only have this single domain. Okay, so speaking of the immunology and mouse genetics, and you know this antibody has this like heavy chain and light chain that's divided into the variable region and constant region. So what we did, how we come up with the idea is to really to humanize the entire mouse Ig locus with human. It sounds easy, but it's not. So because of the different VDJ segments, you can see its entire length is about megabase long. So we took out the task of humanizing entire Ig locus of mouse and replace it in situ with human. And so we start with the heavy chain, and you can see that at the genomic level, it has 68V, 25D, and 6J, but depending on whether you count pseudogene or not. So but we put entire length of the human into the mouse, and so but then we also keep the mouse constant region because we found it is important to maintain some of the downstream signaling. And then we humanize the kappa light chain. And so we did that by put variable region of the human kappa chain into the mouse. So it is in this mouse, the variable region of the kappa is humanized. And we also take on the task of humanizing lambda chain. This is not an easy task because in the mouse, because in human, the kappa to lambda ratio is about 60% to 40%. But in mice, it's about 90 to 10. And it's more difficult to humanize the lambda chain. And you can see this is because, as you can see, that the VJ is clustered in the kappa, but in the lambda, you see that the J segment is kind of juxtaposed to the constant region. So you have to humanize the entire full length, not only of the variable region, but the constant region. So, but we did it because we want to make sure that by maximizing the antibody diversity gives the customer the best chance or best opportunity to for allowing us to identify optimal therapeutic antibodies. Because for antibody discovery is really a game or odds. So by increasing the odds of producing the potential antibodies, you're more likely to get a successful antibody from the campaign. So because it's a mega-based gene knock-in, so we first wanted to make sure that the B cells can develop normally in these mice. So we first started looking at the spleen, and you can see that the T cell NK macrophage is so the purple is our humanized mice, and the black is the BAC6 control. And you can see that the in the spleen, the mice develop normally with pretty much the similar distribution of B cell, T cell, NK cell, and macrophage. And looking at the B cell specifically, we find a small decrease of the mature B cell and the slight increase of the transitional T cell. And our hypothesis was that because of the larger fragments of the human VDJ you put in, so like the VDJ recombinance takes longer for it to happen in this humanized mouse than in the wild type. So that's we see a slight decrease of the percentage of mature B cell. But if you look at the IgG switched B cell in terms of making a secreted antibody, but we found it the same, either under a naive state or under a immunized state. So we just first wanted to make sure the mouse can develop B cells normally. And then, because I said that one of the biggest advantage or competitive advantage what we have is by allowing the maximum diversity. So when we look at the diversity of this humanized mice, so first we look at the heavy chain, and you can see the diversity distribution of the V, D, and the J segment. And you can see that in the normal mice, the CDR3 length is about 10 to 12 amino acid, but in human, it's about 16 to 8. And then we look at our humanized mice, the CDR length, which is critical in terms of determining antigen specificity. That's already, we already skewed the CDR3 length from mouse to human. So it's pretty much overlapping with human. And then we look at the preference of each VDJ segment, and you can see that it's pretty much the same pattern in our humanized mice compared to human. And then looking at light chain is the same pattern. So look at the diversity. So first we look at the VJ in the kappa we have found a diverse distribution of the V and J family, and the same can be said for the lambda chain. And looking at the preference of the VJ usage is also pretty similar to human. And then we went on to develop a series of immunization campaigns to looking at the antibodies raised in this antibody humanized mice. So this is a sort of a schematic drawing of our typical immunization campaign. As Lei mentioned, it's about like 2 months of immunization. And so we tested different targets with different immune strategy. So in target A, it's a recombinant protein, and target B is an mRNA, and target C is a peptide. And what you can see is that really the antibody raised in the humanized mice is overlapping, or you can see that these 2 lines pretty much superimposed to each other in terms of the antibody response. We don't use any sort of special adjuvant. This is just normal the normal immunization campaign strategy, and the same for mRNA immunization. And sometimes, to our surprise, we even have a better titer in the humanized mice than wild-type Black6. And then we look at the diversity of the antibodies or the sequence of the hits we got from the immunization campaign. So this is by hybridoma. And so you can see that it's not against this dominant epitope. You can see that by looking at the distribution of the diversity of the different VJ families, as well as the diversity you can see here in the CDR3 lens. So we were really raising antibody against the different, our hypothesis, this is most likely against the different epitopes of the recombinant protein. And then we then went on to more of like in vitro validation, how this our top leading sequences binds to PD-L1 compared to reference drug or benchmark, that is olizumab. And you can see that in the in vitro binding assay that it has pretty much the very similar binding affinity to the benchmark. And you can see that this is confirmed by BLI. That's also in partnership with Biointron. That the affinity is at 10 to the minus 10s and 10 to the minus 11s. So this is because we really wanted to sort of validate this platform. So we did not only the binding assay, but we also did the in vitro and the in vivo functional assay. So in this in vitro functional assay, it's the first one on the left is the from mixed leukocyte reaction. And you can see that, so this is our leading antibody, and this is the benchmark, where you can see that with the titration of the antibody concentration, you see the inflammatory cytokines decrease, which is indicates the function of the PD-L1 antibody. And so we then went on to do in vivo study to see whether you can inhibit tumor growth in a xenograft model. And we found that it's in terms of TGI or tumor growth inhibition, is pretty much similar to the benchmark. Okay, so this is a summary because we are not only the antibody humanizing providers on the market. So this is like more of a competitive landscape of the humanized antibody you can find on the market. So here that we use a method of ES cell targeting. So it does not, we don't use touch CRISPR at all. So there's no potential liability of CRISPR related to license. And in addition to that, because we created this mice under the Black6 background, but there is this myth that the titer in BALB/c is higher than in Black6, but we don't believe that because what we found is that this is very antigen specific. So we offer the same antibody humanized mice on 3 different genetic backgrounds. And if you're doing an immunization campaign with us, with the partnership with Biointron, you can actually integrate the 3 different genetic backgrounds against the same target. So you get the maximum diversity and the likely chance of a higher immunogenicity or immune response. And as I mentioned before, we are the only company on the market not only humanize the heavy chain, but as well as the kappa and the lambda. So my next few slides is really just showing the partnership of us with Biointron. So by partnering our immunization campaign by Biointron's app job, how we can sort of accelerate the antibody discovery from typically 2 to 3 months to less than 1 month. So this is again, we use the PD-L1 we used before, and you can see the serum titer increases with increasing serial of immunization. So what we did is that by using Intron's platform of AB job, so from one single mice, we collect about 2 million CD138 enriched plasma cell. And then that goes through this process. So what we end up is having more than thousands of pairs. That means it has both heavy and light chain. And it's with the Pupil app that actually 400 unique pairs. So the unique sequences and with the wild type, we have 100 unique sequences from the from the AB job platform. And then we took 20 of the top 20 of them for expression. And you can see that this is the binding affinity. And you can see this is pretty much 15 or 8, close to 80% of the leading sequences from the Hugo app produce the anti-PD-L1 antibody at sub-nano molar range. And to simply have this positive control, and actually we can see for some of the leading candidates, it has a binding affinity of better than the control antibody. And then we also did this with the same strategy we did before. We look at the cell binding and the blocking in vitro. So I'm not going to go through this again, but again, we eventually with the partnership with AB Job, we shortened the antibody discovery from 2 months to 1 month. And then also looking at the diversity. So here you can see this is from our Hugo antibody, and then this is from a wild type. So essentially we're seeing that each line it indicates antibody sequence. So the more scattered the lines, the more diverse your sequences are. And you can see that in the Hugo app and both the normal or wild type control, you got this diversity of the hits from this partnership. Okay, and then because our founder Lance is very focused on or have a great interest in AI, and it's because one of the questions we are often being asked is that we have the best antibody engineers. Why don't we just sort of do humanization? And then, so what we did is that we have this training data set from 1, 2, 3, 4, 5, 6 different species with a total sequence of 1 billion. So we train the AI to know which one is human, which one is camel, which one is mouse. This is our training data set. And then we use a testing data set of 10 million antibody sequences publicly available. And so after the training, the AI model, so this is the algorithm we developed ourselves, and with the AI, we can pretty much achieve 99.7% accuracy in terms of determining whether this sequence is from, say, camel, human, mouse. So what we did is that in that immunization campaign, so we have 139 sequences from the wild type mice. It has so the higher the score, the more human-like. So you can see that this is pretty much close to 0 from our wild type black 6 mice. And then this is all the sequences against the PD-L1. And then we found with the clinical stage PD-L1 sequences on the public database. So you can see that in the humanized mice, even though you said it is humanized, but then in terms of how human it is, it's only 0.6% or 0.6 or 63%. And also in the fully human and in our Hugo app is pretty similar. It's close to 100% human. And then this is another case that we partnered or did with Biointron. So we use another target, popular target in both oncology and the AMD. We use the same strategy to immunize the mice and go through this AB job. And eventually we found close to 300 unique pairs. And what you can see is that the 13 out of 20 of molecules binds to VEGF at the sub-nanomolar range. Okay, the last but not least, so this is actually a customer that come to us and Biointron because they did this project, and because the antigen itself is 90% to mouse, so they use our services and use hybridoma before, but we only to find the two binders and with a period of 6 months. So that includes the immunization as well as the antibody discovery. So that's 6 months. But then because there are only 2 bindings we found with the hybridoma, but so because at that time we just started to partnership with Biointron. So what we give out this platform a try, and from this you can see that from one immunized mice, we were able to get 34 unique pairs, and then we found 28 of binders has a binding affinity similar to the benchmark. So it not only accelerates the antibody discovery process, but also giving you a better chance of screening and getting the antibody sequence you need. Okay, then I'm switching gear a little bit towards the other platform we are currently offering, what we call Hugo Light. So as Lei mentioned, there are like more than hundreds of ways you can make a bispecific antibody, but the most human-like is the sort of what we call common light chain. And so it has one arm binding to one specific antigen and the other arm binding to the other antigen. And the structure is mostly most resemble what the natural IgG looks like. So what we did is that we look at the light chain sequence distribution from the human naive pool, and we see which are what are the top 8 sequences mostly used by human. So we selected the light chain sequences based on its frequency. So we came up with this suite of 8 different common light chain donors. So, and the reason we do that is that we're not only just selecting, like say, 139, 320, that's the most popular ones, because we believe that certain light chain has a preference with pairing with different heavy chain. So by offering this sort of suite of different common light chains, customer can choose and pick which light chain, and actually they can do multiples from one immunization campaign to look at which light chain gives the best possibility of finding a good binder. And so this is again we did because at that time we saw because the single light chain is fixed. If the light chain is fixed, it might compromise the antibody affinity. So we choose the same target we did for the conventional antibody VEGF, and adopted the same immunization strategy. So you can actually see that in the wild, this is our wild type mice, and this is our Hugo mice. So that's why I say at the beginning. So for some of the immunization campaigns, what we found is that actually in the antibody humanized mice, we get a better titer than the wild type control. So partnering with the AB drug platform, and from this immunization, we got close to 200 unique pairs, and we selected the top 20 for expression, and you can also see that. So if you remember from my last slide, the binding affinity from the conventional antibody is at sub-nanomolar range, and also with the single light chain, we produce the binding affinity at similar level. So the conclusion is that by having a fixed light chain really doesn't compromise antibody diversity or the affinity. Okay. So this is a product we currently still developing. I can mention at the very beginning because of versatility of nano antibodies. So we wanted to sort of create nanobody or VHH in the mouse, and the way we did it by knocking out the mouse CH1 domain, which is essentially in partnering with the light chain, as well as knocking out this kappa and lambda chain. So we are sort of forcing the mouse to make an VHH only or single chain only antibody. So this mouse is developing, and we currently have some validating data. If you're interested, you can share after this talk. Okay, and because this this is a very IP-rich environment in terms of making the transgenic mice, and even before we started to make the mice, we look at all the patents on the market and did a very thorough FTO, and so we make sure that there's no conflict and we are free to operate, and so we have three different patents applied for the Hugo map, and two are in the process. And we have FTO for these three different platforms in China, US, Europe, and Japan. So what are the advantage of having our antibody humanized mice? So the first one being a sort of really, you are accessing a platform, not just one single antibody humanized mice. The way I mean is that we have these three different platforms for both the conventional antibody discovery, the bispecific antibody discovery, and the nano antibody discovery, as well as that we offer all of these three on the three different genetic backgrounds. So you can choose, or you can do everything in one single immunization campaign. And then the other thing that sort of prevents the access or makes a hurdle of using antibody humanized mice is that it is a really sort of royalty-based or milestone-based payment scheme for most of the current platforms. So you pay for the mice and then you pay for the immunization. And depending on which milestone you hit, you pay different royalties and milestone-based payment. But we want to make it simple. We want to sort of lower the bar, being a game changer in this environment. So with us and by the partnership with Biointron, you only pay once for the mice and immunization, and that's it. There's no strings attached to it. And we also, because we are a company sort of really focused or specialize in genetic engineering, so we have what we call the EKO, which stands for express KO. So for those high homologous target, we can because we have ES cell stored on the early stage, so we can easily knock out the target and then increase the immunogenicity of the target that you're interested in. Okay, so and again, this is a Biointron event, and thank you for inviting us to this event. And believe through this partnership, we can make this antibody discovery platform more affordable and have more experience. So, and we also have this online platform we call it AppSeek. People can call it abc.ai. For some of the humanization, that's just like one module that's integrated into this platform. But we also have like in simple affinity maturation and the sequence comparison. All that you can try out. Okay, so my last three slides so just a little introduction about our company. So we are a company really focused on animal model generation. So the company is founded in 2006, and since 2008, we're starting to do customized animal model generation. So, and we call this total knockout. So that's when we started to do everything on ES cell. So even though CRISPR was introduced in 2013, and but because we especially for commercial customers, we have the capability to do genetic engineering with CRISPR. For commercial customers, that's sometimes a concern. So we sort of we retain and sort of make the ES cell genetic engineering full blown by now. And right now, we already start, because we're also establishing the knockout as a repository. So if you have any interest in, say, for a particular gene, a knockout or conditional knockout, most likely you'll find it in our repository. And then in 2021, we started doing the CRO, so not only on the antibody discovery part, we also do in vitro and in vivo pharmacology. And so the antibody mouse platform that I just talk about was actually not introduced not too long ago, and in China, and we're already seeing great customers either by partnershiping with Biointron or ourselves. We're seeing great customers starting to adopt into this antibody humanized mice, and but in the US or in the overseas market, we just starting to get more engaged with the customers. And so some of our core solutions, which is as I mentioned, is really the rapid and cost-effective model generation services. And then we also introduced the Hugo GT, So it's the same idea with genetically engineered humanized mouse models, but that's more focused on or suited for gene therapies, and we'll provide the CRO services. And at every step of the way, we try to integrate AI to our services. So we are a company with 900 employees. We have 3 production sites in China. One is in Wuhan, is near Beijing. That's where I am based currently. And then the other one is Suzhou, near Shanghai. And we have also our CRO services in Guangzhou, and have headquarters in Santa Clara, in Bay Area in the US, and have a Japan office. Okay, so this is sort of summarize how we do it and why we do it. So we wanted to sort of accelerate drug discovery with data, algorithm models, and more importantly, with partners like Biointron. So, and thank you again for listening. Thank you. That's a good question. So we currently more focused on the sort of the first part, which is the cell part in terms of developmentability. So we don't find any issue, especially with our sort of Google map, because the antibody itself is of the conventional shape, and for the common light chain is the same. That is in this antibody natural shape, and for the Hugo nano, the nanobody, we don't have the data yet. That's something you're also interested in to know. The heavy chain is not in whole, and then by having the fixed light chain, you really reduce the chance of heavy and light chain mispairing. But the heavy chain pairing is not at fault. Thank you.
All right. So thanks, Jimmy, for your great talk. And now it's my pleasure to introduce Dr. Hitesh Soni. And he's now the director of in vivo pharmacology at Tectonic. And he's been doing a lot of GPCRs, I can say. So that's my old flame. I'm still working on GPCR targets, and that's it's been my passion here. And he's got a PhD Gujarat University, and also an MBA from the University of Memphis. And he's wow, that's also with a perfect GPA. So and not only that, he's obviously a academic researcher with a lot of NIH and the AHA grant contributions. So welcome, Dr. Soni.
Thank you for the nice introduction. I'm not an immunologist, I'm a pharmacologist, and I'm going to talk about something which is more applicable in real world. So these guys talked about how antibody can be developed and then how we can use as a tool. And we are one of the customer for the Biointron, and we are using their platform as well. We are generating a lot of antibody in-house, but also for the bulk production. I'm a macrophage in our Tectonic, generally a big eater of the protein. So generally I ask for large quantity for the animal studies. And Biointron is one of the service provider. They're helping us getting more quantity to us. So, okay, let's get started. I hope nobody's feeling sleepy. So I hope I can keep the spirit so that you cannot fall asleep. So it is truly an honor to be here at the Biointron Lunch and Learn series. And I would like to thank the team Biointron for the invitation. I'm going to talk about one of our fusion protein, FC fusion protein. It is called TX45, which I would say it's a kind of best-in-class molecule so far. And we are trying to develop this molecule for the treatment of group 2 pulmonary hypertension, which is the condition of hypertension associated with the lung. The typical symptoms is dyspnea. The patient has a problem breathing. So we are developing that molecule, and I would glad to say that the molecule has already passed phase 1A and 1B trial. It is moving to the phase 2 trial now. But today I'm not going to talk about all those clinical stuff. I'm only going to talk about the entire history, like how we bioengineer this molecule, what were the trouble associated with that, and then how we develop this molecule using our preclinical platform. So I'm basically going to talk about that. But before we go into the scientific part of it, I would like to start with one of the quote from Sir William Osler. The good physician treats the disease. The great physician treats the patient who has the disease. This is a great philosophy, which is mostly the patient-centric approach. And this philosophy resonates with our goal of transforming patients' lives. That is exactly what we are trying to do with our molecule called TX45 for the treatment of group 2 pulmonary hypertension and this molecule is basically FC relaxin fusion protein. I need to put this slide anyway, because I'm from the company. So this is a standard disclaimer slide. You don't need to read about it. There are a lot of text into it, but I have to show that. Before we jump into the actual data part of the entire journey of the molecule, I don't know how many of you are aware of the relaxin as a hormone, which is a pregnancy hormone. Any one of you know about this hormone before? Anybody? I saw somebody is nodding head. That means someone is knowing about it already. And hopefully many of us, many of the other people not knowing about this hormone. So if you are knowing, that's great. Like this is going to be a refresher. If you are not knowing, then I think I will try to convince you that it doesn't have only the function related to the pregnancy. It has also other functions as well. So let's get started with the basic part. This hormone, relaxin, is basically a potent pulmonary and systemic vasodilator. So it is important for the pregnancy adaptation. And basically, it increases the cardiac output to accommodate the increase in demand from the developing fetus. That's one way. It is potent vasodilator. The other function of this hormone is related to the fibrosis. This hormone is also anti-fibrotic. So basically, it prepares the muscles for the pregnancy and childbirth. So that's very well-known function associated with this hormone. If we go back to the pharmacology part, and as Dr. Shi also worked with the GPCR, so basically this hormone acts through class A GPCR. It's called RXFP1 receptor. So relaxin is basically natural ligand for the RXFP1 receptor. This is class A GPCR, and I would say that this is a unique GPCR. Why I'm saying unique GPCR is because unlike other GPCRs, such as adrenergic receptor or opioid receptor, upon activation, this receptor does not go internalization or desensitization. This is very important function when you develop the drug for the chronic diseases. And that's why this is one of the good target for the disease, such as pulmonary hypertension or heart failure. The other important fact about is that the relaxin hormone is upregulated in pregnancy. But I keep talking about the pregnancy hormone. So you might be wondering that whether it is also there in the man or not. And I would say that yes, this hormone is available at a very minimal level. It is very important for the reproductive health. So that's why it is available in man and women as well. The levels in man and women, which is a baseline level, is around 50 to 60 picogram per ml, which is very low level. However, during pregnancy, these levels go very high. In first and second trimester, the levels of the relaxin hormone goes almost like 1 to 2 nanogram per ml, which is, I would say, like 60 to 100-fold high during pregnancy for the adaptation for the childbirth. So these are like important fact about the hormone. The other important aspect about this target that this molecule, such as relaxin and other mimetics, one of the molecule which is there in the literature, it's recombinant protein for the relaxin hormone developed by the Novartis. So these hormones are active through multiple mechanisms of action. And you can see here in this cartoon is that this upon activation, it acts through the endothelin pathway. It also acts through the nitric oxide pathway. And it also acts via TGF-beta signaling pathway. So the signaling mechanism, it has a multi-mode action. However, the standard of care therapy for the pulmonary hypertension for the group 1 patients is sildenafil, which is a PDE5 inhibitor. And you can see that act only through nitric oxide pathway. There are prostacyclin analogs that are also available in the market, and they also act through vasodilator. So we thought this is very unique target, which acts through multiple mechanisms. The other interesting part about the pathophysiology of the group 2 pulmonary hypertension, where these all 3 mechanisms are involved. And the other important part about the group 2 pulmonary hypertension is there are no FDA-approved drug available for the patient. So several other facts were also available in the literature. And as I said, like one of the recombinant protein, it's developed by the Novartis. It's called serelaxin. So serelaxin, when given at 30 micrograms per kilogram per day, as an infusion, and you can clearly see in the right side of the graph is that it was clearly reducing the pulmonary vascular resistance in acute heart failure patients. So we knew from the beginning is that it has an impact even in the patients. The other important fact from the clinical trial data, you can see in phase 2 clinical trial, the phase 2 clinical trial is known as pre-relax AHF and relax AHF. It was fantastic trial. You can see here the serelaxin administered at the 30 microgram per kg per day infusion for 48 hours. It demonstrated 20% improvement in dyspnea through almost like day 5, which was like amazing data. And then it got like fast track approval for getting into the phase 3 trial. However, this is unfortunate part is that in phase 3, which is a very large trial where they use almost 6,000 patients, this did not meet the primary endpoint. And the primary endpoint was very tough, like reduction in 180-day cardiovascular death using the infusion for 48 hours. So unfortunately, the trial has been failed, but there was a lot of learning from the phase 3 clinical trial. And another interesting literature you can find about the meta-analysis. Once the drug has not been approved, somebody has done the meta-analysis and published. And it indicates that when they combine all the 6 randomized controlled trials, serelaxin reduced the incidence of worsening of heart failure by day 5. And you can see the p-value was highly, highly significant. Imagine that this could be the endpoint for the phase 3 trial, that this drug could be helping the patient by now. So there were a lot of learning about this trial as well. And from all this information, we were very clear about our aim is that we are going to develop the potent relaxin agonist, and we are going to create the molecule which can help with group 2 pulmonary hypertension patients associated with the left heart disease. Okay, this looks easy. So the initial challenge we found is related to the native human relaxin, that it has short half-life. And therefore, it requires like continuous IV infusion, which limits its therapeutic application because there is no patient compliance if you keep giving the intravenous infusion. So the simple approach, you can just fuse this hormone with the FC domain of human IgG1, and you can put on half-life. Pretty simple, right? And the other part is that we did a couple of mutations in the FC part. So we did the FC silencing approach because we don't want any effector function. So we did the silencing of the FC. We also did some mutations where we can increase the binding to FcRn. Like FcRn is basically a neonatal FC receptor, which are important for the recycling process so that your half-life is going to be increased. So that's what we did. And we linked to this hormone. This hormone is similar to the insulin. Like it has A chain, B chain, and we linked this molecule with the linker. So again, like we thought that, oh, this easy formula is going to work. It did not. You can see here that our early molecule, it's called PX04. And the structurally different molecule, relax-10, when we wanted to, because our first aim is to enhance the pharmacokinetic profile. So we used this molecule and ran the PK study on rat. And unfortunately, you can see there was almost like 90% decline in the alpha phase of the pharmacokinetic curve within less than a day. Both the molecule was showing like suboptimal PK. And one thing was common. So we tried to dig deeper and we tried to understand that even though these molecules are structurally different, why they behave similarly. And we found one property which is common between these two molecules called isoelectric point, which is PI. I would like to simplify this. Like isoelectric point is basically stickiness of the molecule. As you go far from the pH of the body, your molecule becomes more positively charged and sticky. If you reduce and go towards the pH of the body, your molecule becomes freely available. We hypothesized that these high PI molecules might be binding to the negatively charged heparin sulfate proteoglycans and have poor PK properties. So again, like we wanted to use some approach, which is the engineering approach, bioengineering approach. So what we have done is that we have strategically reduced the positive charge of the single chain relaxin Fc. Now here, the engineering part was very critical that the native relaxin has many arginines. Now these arginines are positively charged and they make the molecule more sticky. So, the other thing is that the arginines in the B chain, some of them are very important for the binding. So arginine at position 17 in B chain and position 13, we did not change because we know from the literature is that these are very important for the binding. However, we did a couple of changes. We neutralizes the charges using some mutations in A chain as well as B chain. It also has a couple of histidines. So we also replaced those histidine with the other molecules. And I'm not going into the detail of what we did actually. But the other thing we did is the linker. So we used a very short linker between FC and B chain. And we also used a linker, which has a neutralized charges and negatively charged amino acids. And overall, we try to reduce the PI of the entire molecule using this FC fusion. And the results are here. You can see here. So as I showed these two molecules, the high PI molecules have suboptimal PK by improving the PI. And I would like to say exactly the 7.8 PI for the molecule PX45. And we believe that this is a kind of PI of which we believe that it is a Goldilocks zone of the PI we found for this molecule, and it improved the pharmacokinetic profile. So after improving the pharmacokinetic profile, next we wanted to know that this molecule is potent enough or not. So we used in vitro approach, and we wanted to see whether TX45 has a low nanomolar potency or not. We used the HEC293 cells, which transiently express human RXFP1 and rat RXFP1 receptor. And you can see here that we could able to maintain the low nanomolar potency. You can see the EC50 against human RXFP1 is 10 nanomolar, and EC50 against rat RXFP1 is 30 nanomolar. So we were pretty confident that this molecule is potent enough, even though it is not as potent as the native relaxin, but we at least improve one property. As you keep improving the pharmacokinetic profile, and if you keep lowering your PI, your molecule becomes less effective. But we were very sure that still it is a good potency overall. And you can also see here, interesting part, the comparator high PI molecule in this in vitro assay, specifically, it was more potent as compared to our TX45. So we took that as a challenge that let's move forward and let's see what happens to the next part. Before we dig into the in vivo studies, we also wanted to make sure that this is more selective. So we did the selectivity study using stably expressed CHOK1 cell line, which expresses the human RXFP1 and human RXFP2, because we wanted to make sure that it doesn't touch the RXFP2. RXFP2 has other functions, like it is most important in the reproductive health. And also in the bone formation, it has some role. So we want to make sure that our molecule is more specific towards the human RXFP1. And we did the in vitro assay, and we found that it has pretty much low nanomolar potency against human RXFP1 and multiple fold increase in the EC50 value against human RXFP2. So next, we wanted to see that how in vivo these molecules are behaving. And we wanted to see that what is the pharmacodynamic behavior of this molecule. One of the models we used is for rat renal arterial blood flow model. And this is the cartoon which indicates that how we did this in vivo studies. The other interesting part about the rat renal arterial blood flow is that during your phase 1B clinical trial, you can also see the vasodilatory effect in the healthy volunteer itself. So early enough, you will come to know that whether your molecule is working in the humans or not. So this is a kind of translational approach we took. And you can see that rat has been anesthetized with the urethane. And then we isolated the renal artery, and we implanted the flow probe and attached with the flow meter. And we measured the blood flow for almost 3 hours. And the initial PD validation studies, you can clearly see that our low PI molecule performed better compared to the high PI molecule, initial molecule at the same dose. So this was giving a validation that yes, our PI hypothesis is working, and the molecule is also superior compared to the initial molecules. It could be because of the pharmacokinetic profile. So the other interesting part about this experiment, in vivo experiment, is related, like I can simplify this in this way. So as I showed that the comparator molecule, which is a high PI molecule, in vitro assay, it has 10-fold higher potency. And in terms of exposure, our molecule behaves almost 10-fold better. So in theory, if we use this molecule for the PD validation at the same dose, you expect that it behaves similarly. However, our TX45 molecule exceeded our expectation, and it was almost like 10-fold more potent compared to our comparator molecule in vivo studies, which gives us more confidence about the PI hypothesis, that we found a Goldilocks zone of the PI, which improved not only the pharmacokinetic profile, but the pharmacodynamic behavior of the molecule. So after seeing these fantastic results, like with the pharmacokinetic behavior and a PK-PD correlation, we wanted to see whether this molecule can do something with the disease model. But before I go into that, we also did the dose response study and using the same model, renal arterial blood flow model. And you can see here that the TX45 dose dependently increased the renal arterial blood flow, and there is a linear pharmacokinetic behavior of the molecule. The other interesting part when we did this dose response study is that our in vivo EC50, when we derived from this curve, it was 34 nanomolar. And as I showed you that in vitro EC50 against rat RXFP1 was 30 nanomolar. So it was beautifully aligned very well with this study. Then we moved to the in vivo part of it, like disease model, because I think that is the ultimate goal, that whether the molecule is working in the disease model or not. Again, like nothing is without any challenges. There was another challenge here. We developed the human FC of IgG1. If you use in rat for the chronic administration, there's a risk of ADA. So we know that there could be the ADA risk. That was one problem. We also found the literature where it has been proposed, or somebody has done the study where they have used the human relaxin 2 infusion using the mini pump in the rats in the same model. And they found that it was causing the generation of a neutralizing antibody, and there was no efficacy in that model. So this was alarming to us, and we wanted to mitigate that risk. The other problem is that we did a lot of validation studies because there was no great antibody available which cross-react with the rat, which can deplete the B cells to reduce the ADA risk. However, we defined our protocol, and we used the rat cross-reactive anti-mouse CD20 antibody. The protocol is very aggressive here. But we wanted to make sure that we can deplete the B cells. So you can see here that initially in day 8, 9, and 10, 3 times initially, we need to give this antibody at 20 mg per kg. And then we need to give 1 maintenance dose and we validated by flow cytometry that we are depleting the B cells and we don't see any, or we see the less ADA response. And briefly, I can explain this model because we use the model which is called generating the pulmonary hypertension phenotype. It's called monocrotaline-induced pulmonary hypertension in rats. So this monocrotaline, which is a kind of toxic material, which leads to the damage of the pulmonary artery, the endothelial cells of the pulmonary arteries, and it causes the disease phenotype. I will go into the detail in the subsequent slide that what kind of disease phenotype it generates. But it is very consistent across the literature is that this model is very robust and people have used it for the screening of these kind of molecules. So briefly, what we have used the male Sprague-Dawley rats and we gave the single injection of monocrotaline at 60 mg per kg subcutaneously. And we use our molecule in therapeutic mode where we started the treatment a week later, twice a week for 3 weeks. And then we terminated the study by day 28. We performed the hemodynamic measurements, and then we collected the blood for the exposure assay. We did another endpoint. It's called Fulton index. Basically, it indicates about the right ventricular hypertrophy in the heart. And then also we collected the lung samples for the histology. Results were very promising. So you can see here in the left side of the graph, which is a Kaplan-Meier curve, of the survival rate. And you can see that with the treatment arm, we got almost 100% survival in 4 weeks of, like 3 weeks of treatment and total duration of the 4 week, with respect to like 70% survival rate in the disease condition. So in disease, you will find 25% mortality, whereas we didn't find any mortality with our treatment arm. The right side of the graph is indicating about the right ventricular hypertrophy. We measure by the index called Fulton index. So Fulton index is basically the ratio of the right ventricle versus the left ventricle plus septum of the heart. And you can see here that the monocrotaline induces the right ventricular hypertrophy, and which has been significantly reduced with our treatment arm, which is TX45. This is another parameter we measured. It's called right ventricular systolic pressure and mean pulmonary arterial pressure. If you see the literature, the group 2 pulmonary hypertension patients has been divided in a different types based on the type of pressure responses in their heart. One of them is mean pulmonary arterial pressure. And if you see the group 2 pulmonary hypertension patients, they have MPAP more than 20 millimeter of mercury. So that's why this is very relevant. And the gold standard for the patient is also a kind of pressure measurement where you do the right heart cath, which is called RHC. And then you measure the actual pressure in the heart. And if your treatment is working, you can see the reduction in the pressure. So the similar approach we used in the animals also, and you can see that right ventricular systolic pressure. And I don't want to go into the physiology part, but if you see the right ventricle is basically a volume chamber, the wall of the right ventricle is very thin to accommodate more volume. It's like balloon. However, during the when you give the monocrotaline, the thickness of the right ventricle is going to increase. And that also, so to adapt to the pressure, it increases the thickness and hypertrophy, and it also increases the pressure as well. So it is called vascular remodeling. So this is a remodeling effect happening in the right ventricle. And you can see there is a almost 3-fold increase in the right ventricular systolic pressure. This has been almost like 40 to 50% reduction with the treatment arm with the TX45. When we measure the mean pulmonary arterial pressure, so when I'm talking all this, it looks very simple, but in rat, you might be knowing that the heart rate of the rat is around like 300 beats per minute. And under anesthesia, it goes a little bit down, like 250 beats per minute. Not unlike human, it is like 72 beats per minute. And putting the probe inside the heart is one of the challenges. which we developed everything in-house. And then we could, and when there's a high pressure, when you put the probe inside, it tried to come out because of the pressure. So, but we did everything in-house. We developed the technique that how we can do it. And you can see that our molecule is significantly reducing the mean pulmonary arterial pressure after treatment. So we collected the lung samples, and this is also one of the hallmark feature of the pulmonary hypertension. One part is called plexiform lesions, and it also generates the muscularization of the small pulmonary arteries. When I say muscularization of the small pulmonary arteries, is basically there is an endothelium damage, and then the smooth muscle cells start proliferating, and then the vessel becomes narrow. So we wanted to see whether there is change in the, so this condition is called vascular remodeling effect. And you can see here in the right side of the graph as monocrotaline is increasing the vascular remodeling part by increasing the hypertrophy, medial hypertrophy due to proliferation of the smooth muscle. So this is based on the diameter we have measured, and we can see that there is increase in the hypertrophy part or the length of the medial part of the artery, which has been significantly reduced with the treatment arm. We also wanted to see that what happens to the lung overall histology score. So it has been significantly increased with the monocrotaline arm, and it has been significantly decreased with the treatment arm. This sum score also includes a lot of inflammation criteria as well. So we can also see that not only the vascular remodeling happening, but also the inflammation part has been reduced with the treatment. So to summarize everything, like we did a smart engineering approach, and we developed the best-in-class molecule called TX45. And we believe that this molecule has great pharmacokinetic behavior and also pharmacodynamic profile. This was another interesting observation that in rat RABF in vivo model, when we calculated our EC50, that was 34 nanomolar, which was beautifully aligned with the in vitro potency of 30 nanomolar in rat RxFP1 assay. Next, we also tested this molecule in the disease condition, and then we found that there was improvement in the hemodynamic. It reduces the lung inflammation, and there was a marked reduction in the arterial muscularization and vascular remodeling. So we believe that these positive changes has created the beautiful Kaplan-Meier curve, Kaplan-Meier curve, and that's why we could able to see the 100% survival. So these optimized properties position TX45 as a promising therapeutic candidate for group 2 pulmonary hypertension. And I would like to express, okay, so before I get into that, I wanted to circle back to the original quote from the Sir William Osler, is that with the TX45, it's not just about treating a disease, but to transform patients' life. With that, I would like to express my gratitude to our C-suite, Elise, Dan, Peter, Tony, Marcy, and Mark. I would also like to thank our co-founder, Tim Springer, Andrew Cruz, and special thanks to John Diener, whose strategic thinking was very instrumental to make molecule TX45. And also entire Tectonic team, their love and support was very important to get to that stage. And I would like to thank you for your attention and time. I'm happy to take any questions. Thanks. So we were using surrogate for the heparin sulfate binding, which is called heparin binding for the molecule. And we tried so many molecules for the pharmacokinetic behavior with the different PI. And we found really like nice pharmacokinetic behavior, especially in terms of alpha phase with the TX45. So we tried a couple of molecules during our PK validation. This PI stand out well in terms of pharmacokinetic behavior in rat assay. What was the other question? I don't think so that is unique to us. I would say like people are using this strategy, and you can find in literature as well, various literature, yes. Yes. No, there is no PEG, no albumin. We are not using any of this. We are using purely the human FC of IgG1. And we are also mutating that site. I think there is a famous mutation. It's called LALA mutation. When you do that, you knock down the effector function. There is another mutation, LALA PALS. So LALA PA is basically reducing the effector function. PS mutation is important for the FcRn binding. So when you do the mutation, it enhances the binding affinity towards the FcRn. That's how you can increase the pharmacokinetic behavior. That's a good question. Yeah. So we used another model for the fibrosis. It's called unilateral ureteral obstruction in mouse. And we found the anti-fibrotic effect as well. Yeah, that's correct. Yeah. Question? Good. I hope you understand very well. Thank you.